Hello everyone, I'm Cushing. I'm a fourth year PhD student working with Yuri. So today I'll talk about our project on using this graph learning over massive functional genomics knowledge graph to do small cohort genetics discovery. So this is joint work done with an amazing set of scientists from Stanford AI, Stanford Genetics Department, CMU, Arc Institute, and also our industry partner, GSK. So I want to first start with a brief review of what is a human genome. So the human genome basically encodes the blueprint of our body. You can think of it as like a text file of 3 billion tokens of A, C, G, and T, and it basically instructs everything from the cellular functions and all the way to your complex traits like your height, your hair color, and also your health. So not everyone has the same copy of genome, and there's kind of these small differences between from person to person. And these small differences is also known as a genetic variant. And so, for example, here, this is a variant which is usually denoted as this RSID, and it's located at the 55 million-ish position at the chromosome 19, and it has a mutation from the T to become a C. So there's lots of these mutations happening for each one of you, which makes you unique, and also makes you susceptible to potentially some diseases. So for example, this variant that I was talking about is actually a very famous, well-studied variant called E4 allele. This variant will lead to kind of a protein conformation change and leads to increased amyloid beta, which leads to Alzheimer's disease, which is one of the most prevalent diseases in the United States, causing 6.9 million cases. And there's numerous these kind of variants associated with the disease that can cause the disease. So the central task of drug discovery is just to find this kind of right variant for the diseases. The reason why it's so important is because in order for you to develop an effective drug, you first need to understand the mechanism of the disease clearly. And finding the right genetic variant of the disease is the very first step. And it's also a very important step. Actually, one of the leading causes of failure of clinical trial is actually a wrong target. So if you have a wrong target and you spend so much money to develop a drug, you're wasting like 10 years and billions of dollars. So you really don't want that to happen. So this is the kind of central question that we're interested in investigating, that is how to find variants out of these 3 billion options that are associated with the disease. And this is an extremely difficult task, right? Imagine you have a 3 billion character text file, and you want to find a few single typos. It's really like finding a needle in a haystack kind of thing. So this task is extremely important in the drug discovery domain. And there's a existing work that has been done for the past 20 years, and it's called Genome-Wide Association Study, or GWAS. So the intuition of GWAS is actually pretty straightforward. Basically, if you're interested in looking at the one mutation from this C to T at this specific position, you'll collect patients with the disease and collect patients without the disease. And then you basically count the number of patients with the disease, how many of the variants are there in the disease patient cohort. And similarly, you count the number of variants in the healthy patients. And you can really compare the difference between the disease frequency of that variant and the healthy frequency of that variant and see if there's a significant enrichment. And there's a kind of a hypothesis testing framework to produce a p-value that measures the variant association to the disease. So that's kind of what is basically the GWAS. And you will do that for millions and millions of variants. And you will produce this kind of plots where you will see a peak if there's an enriched signal of variant disease associations. So just to be a bit more concrete here, the output of GWAS, it will be a summary statistics file looking like this. So you have basically the important columns here is the ID, which is the variant ID, and then the p-value, which is measuring the variant association to the disease. And because we're scanning millions of variants, usually we do a 5e-assay, like genome-wide significance threshold, to see the variant is like associated, the set of variants that are associated with disease with high confidence and forced discovery control. And another representation is the Manhattan plot, as I mentioned before, where the y-axis is the minus log p-values, and you can see a peak if there's this variant is has very low p-value, for example, below the 5e-assay threshold. Cool. So that's a very brief introduction to what is a GWAS. So GWAS has being applied to many, many diseases of interest, and everyone is kind of trying to apply GWAS to their own kind of drug programs. And in total, it has identified thousands of variants for common complex diseases. And there's also a recent article trying to say the impact of GWAS for drug discovery, and it's estimated that the probability of success for drug mechanism with genetic support is 2.6 times greater than those without. So that's why every kind of pharmaceutical company are extremely interested in this human genetics and have a big department of geneticists. So now hopefully I've convinced you that GWAS is very important and is great. So what is the problem? Why are we kind of want to develop a new method here? So the key problem of GWAS is like it requires huge sample size. There's numerous reasons for that. The two main reasons, like the variant for GWAS is usually have small effect size, so it usually requires a large sample size to detect this kind of tiny variation with high confidence. And there's also, because we're scanning millions of samples, variants, we need a very stringent multiple hypothesis testing adjustment. So this is usually we require a huge number of sample size in orders of tens or hundreds of thousands. But actually, when we really wanted to deploy this GWAS method to the disease of interest, where we find that the disease of interest usually have extremely small sample size. So for example, rare disease. In total, we have 7,000 rare disease, and 90% of them have no effective treatments, and most of them, we don't know the underlying genetic mechanisms, and it collectively impacts like millions of patients. So applying GWAS to this small cohort in, like, for example, rare disease is often have the most impact, but it's also what GWAS currently cannot do. And similar, and I guess other than rare disease, also many common disease also does not have this big cohort because there's cost in collecting the disease sample, and there's data privacy and consent issues. So for these kind of diseases or these small cohort settings, we really need a more data-efficient method for identifying these variants. So just to be concrete, I guess for the AI kind of task, what we aim to do is we take in a GWAS summary statistics. We have an AI model that can produce a new summary statistics, where we will produce a new p-values for each variant, and this new p-value should have same level of forced discovery control, and also ideally should have abundant novel discoveries, especially in the small cohort regime. So now I'll talk about our method. So basically, if you look at the original GWAS method, the GWAS basically scans through the genome one variant at a time, only looking at the genetic information. And because it is, when there's a small cohort, we definitely need to solicit from other kind of information. And recently, there's a burst of this kind of functional genomic screen that mapped the molecular function of a variant. So for example, from variant to gene, there's like QTL studies, there's CRISPR experiments, there's Hi-C data that they are kind of clearly mapped the variants functions to the correct genes. And there's also historically abundant data around gene to genes, including physical interaction, inhibition, binding, and so on. There's also gene to program data like perturb-seq pathways, and so on. So these map the kind of the relational information from that contextualize the variant to the molecular context. And also, there's also variant annotations. There will be like methylation status, chromatin accessibility, and also recently there's a burst of foundation model embeddings that can annotate the variant to give some informative information about the variant. And there's numerous data resources scattered across these different databases to map these variant functions. So intuitively speaking, why would this data help? So imagine if you have a variant, if you know beforehand, this variant is in a region that is known to be functional, for example, it's in an open chromatin region, then we have increased prior that this variant should be associated with the disease. And also, there's also a lot of this relational prior. For example, if a variant is, if we look at the local neighborhood, that's this molecular context of the variant, and we see that there's like a few genes or programs that are known to be associated with the disease, then we also have an informed kind of increased prior of disease associations. So I think our method, KGO, is basically we want to utilize this information to boost the GWAS power, especially in small cohort. So now I will delve into our method, which is called KGWAS, which stands for Knowledge Graph GWAS. So the first step of the method is because basically, if you, as I mentioned, there's so much functional genomics data, but they're all coming from different experimental data. They're coming from different experimental screens. And we provide the first resource to map the genome-wide functional genomics data, and everything, every different kind of experimental data can be consolidated into one place, which is a knowledge graph. Where in the knowledge graph, there will be 3 types of nodes. Like there's a variant, there's a gene, there's also the gene programs. And then there's also this relation which is defined by each individual experiment data. So this kind of represents the first genome-wide variant to gene to program knowledge graph. And in addition to the relational information, we also curate lots of annotations for the variants, including the epigenetics profile, and also similarly for the genes, including single-cell RNA experiments. So given the knowledge graph, we want to now create a method that can distill a knowledge graph prior, which measures the variance prior based on the knowledge graph information. So we develop an objective function to compute this knowledge graph prior. So the basic step is we first train graph neural networks to fit the GWAS association to nodes and edges. So this way, the node embedding and edge embedding implicitly should capture the GWAS association. And then the step 2, we will predict the variant association for every variant based on the knowledge graph information. You can think of it because if you look at the variant, if you look at get the local 2-hop neighborhood, it will basically picks up how much this local neighborhood has enriched disease associations. And this predictive variant association can be used as this knowledge graph prior information or prior score that can be used to adjust the p-values. And the last main module of the framework is a covariate-based p-value weighting framework, because for GWAS, there's lots of, we're scanning millions of variants, so force discovery control is essential. You don't want to create a method that just gives you lots of false positives. So we utilize a statistical technique called a p-value weighting from this paper, where it dynamically adjusts based on the covariates, which is knowledge graph prior, to generate a new p-value, which we call KGWAS p-value. And this KGWAS p-value is used as the new summary statistics of the method. Cool, so that's kind of a brief overview of the method. Now I'll delve into the extensive kind of result that we have. So the first set of results is around the statistical simulations, because we want to show that our method controls the false discoveries. So we first kind of run extensive statistical simulations where we try different configurations of the genetic architectures and so on to see the model's performance at different scenarios. And we see that across the board, we're able to achieve the similar level of false discovery as the base GWAS, while have more discoveries in a causal simulation where, So, but simulation is still kind of a simulation. It has lots of assumptions. So in our case, we were really interested in finding a framework to rigorously evaluate the small cohort, if we can identify more hits, more discoveries. So we leverage a UK biobank, which is a big data set, consists of around 500K patients with raw genetics data and also lots of phenotypes. And what we do is that because we're interested in small cohort, we can basically downsample the large cohort to a small cohort of any size of interest. And then we run the GWAS and KGWAS on a small sample size, and where we have a list of discoveries. And we can check if this discovery can be replicated when you have much larger sample size. So this way we can rigorously evaluate, and also in a systematic way, about the discovery in a small cohort if it's real or not. And we can do that for any kind of a number of traits. So we run that across around 20-ish traits, independent traits, and we're able to see that our KGWAS method actually discovers 90% more, up to 90% more replicable discoveries than GWAS at 1,000 sample size. So this is a significant increase over the base GWAS. I guess on another view, we can also interpolate the number of sample size required by KGWAS to achieve the same number of discoveries as the base GWAS. And we're able to see that KGWAS is actually up to 2.6 times more data efficient than GWAS. For example, KGWAS only required 355 samples to get the same number of discoveries for GWAS at 1,000 samples. So this means that for geneticists, you don't need to recruit such a large number of samples. You only need to recruit 2.6 times less sample size. So this really facilitates the discovery process. So now we kind of established that our KGWAS method is like rigorous and also can have lots of power improvement in this kind of systematic evaluations. Now we really want to apply it to a large number of small cohort diseases to see if we can identify novel genetic insights in a systematic fashion. So we curate a data set of 554 diseases with less than 5,000 cases. And notably, among them, 144 or 25% of diseases actually match the rare disease prevalence. So these are kind of rare diseases where most of them do not have much hits, much known genetic understandings. And you can also see that these 554 diseases covers all the major disease areas, all the way from like cancers or nervous system and so on. Cool. So this is a summary result of our KGWAS applied to this large number of diseases. Overall, we're able to make 576 discoveries, and each of them could be interesting and translates to potentially some drug programs. And this corresponds to a 56% increase over the base GWAS. And you can see that the improvement is like consistent across all the disease areas. And notably, we are very excited to see that actually KGWAS is the most has even more improvement in the rare disease. For example, in this 144 rare disease, KGWAS has 80% more discoveries compared to the average 46% here. And another thing to look at is look at the diseases with these 0 hits in the original GWAS. This means that if KGWAS can find a novel, find 1 hit, then it potentially leads to initial understanding of the disease mechanism of these diseases. And we're able to do that for 92 diseases in this 500-ish diseases. So overall, I think we can conclude that KGWAS greatly expand the genetic discoveries across a wide range of diseases in a kind of consistent fashion for both rare and common diseases. So here I want to look at kind of 2 examples to really show that the KGWAS discoveries are not kind of random. They're actually aligned a lot with the functional evidence. So the first example I'm looking at is ulcerative colitis, which is not a common disease, and there's no effective treatments, and it affects many patients in the US. And for the UK Biobank, there's around 4,000 cases. And we apply KGWAS, and we find this novel variant at this position, which is not significant in the base GWAS, but is significant in the KGWAS. And we go ahead and do this kind of functional evidence research, where we first find that this variant is actually EQTL of this gene. And this gene is actually a known kind of immune gene that can potentially regulate the colitis disease. And then we also, to our surprise, we also find that actually there's an independent study published in Nature 2020 that explicitly knock out the region of that variant and see the phenotypic changes in the colitis. And we find that actually, and they find that this variant actually has significant impact on the colitis histological scores. And I guess another example I want to give is on a real disease, which is called myasthenia gravis. This is a true rare disease, and it leads to lots of symptoms like difficulty swallowing, double vision, unsteady walk, and there's no effective treatments, there's unclear mechanisms, so it's a very important rare disease. And in the UK Biobank, there's 358 cases, and then we apply it to KGWAS. And we're also able to find 1 variant that is kind of missing in the base GWAS, but is kind of significant in the KGWAS. And we find that this variant is enriched in neuron-related cell types, which is a kind of a known related cell type for this MG pathogenesis. And also, this variant is an EQTL of this gene, and we did some research and find that this gene is actually involved in this epigenetic sequencing, but it's complex, and this complex is known to cause symptoms that are similar to this MG disease. So these 2 examples hopefully show that this KGWAS variants, they align really well with the functional evidence out there. So KGWAS actually also have many other downstream genetic applications. For example, because we're using a graph neural networks to fit the GWAS association over the knowledge graph, we can learn a disease-specific knowledge graph We use a graph interpretability or explainability technique to reconstruct that. And we find that this GraphXAI prioritized network actually have overlap a lot with this orthogonal evidence. And because GWAS is so fundamental for the past 20 years, there's lots of methods building upon GWAS. So if we have a better GWAS method, then you can just simply connect with the other method and see if the performance increase as well. So first, for example, we connect the GWAS summary statistics to Magma, and we find that it is able to identify disease-critical genes in a much better, much higher accuracy compared to the base GWAS. And also, we also to connect to SDDRS, which is a method to detect disease-critical cell populations. And we're able to see it can detect novel clusters of cell population that is missed by the base GWAS. And there's more in-depth kind of analysis in our paper. So cool. So that's all of my talk. So we actually released the UI today, which is at kgwas.stanford.edu, where you can explore for your disease of interest. And also we'll release the preprint and GitHub very soon. And I want to thank again for all my collaborators. And thank you.
I have two questions. So the first is, have you guys done any ablations to look at what aspects of the knowledge graph are most useful for getting better performance over more traditional methods?
Yeah.
I know a lot of people in the omics community think that incorporating cell state information from omics might be really helpful, and I'm curious for you guys if that seemed to be a difference maker.
Yeah.
And the second question I have is that a lot of indications or traits in the UK Biobank are really correlated with each other. It sounds like you guys have incorporated knowledge about interactions amongst things like genes. But I'm curious if you guys have played around at all with interactions among the outcomes that you're trying to predict and if it's useful for predicting things for rare disease, right? So it might be that rare disease individuals present with certain blood pathologies that are measured in a lot of individuals in the UK Biobank, and maybe leveraging that information at the level of hundreds of thousands of people can help you better understand those really rare cases too.
Yeah, I think that's two great questions. So for the first question, actually we have done extensive ablation study in the paper. So maybe a few kind of important factors. The first one, we try to permute the knowledge graph structures. So just to try to see if the knowledge graph indeed have this structural information. And we find that if we permute that, actually the KGWAS just becomes similar to GWAS. So that means that the knowledge graph structure, this relational information, is kind of the most important thing. And we also ablate on the individual relation types, and also remove for example, the variant, like variant annotation and the cell annotation, and also try various different alternative variant annotation and cell annotations. And we find that our recurrent version is the most the best kind of combinations basically. And I guess the last thing is we also try to scale the knowledge graph, because we can randomly remove a fraction of the knowledge graph into like 10%, 20%, and 50%, for example. And we'll see that as the fraction goes larger, the performance just goes larger and larger. And I think this also is interesting because it can potentially hint at like a scaling law, like in some sense. If we add, because functional knowledge graph still is very, very vastly incomplete. So if we add much, much more data, it should ideally improve and improve and improve. So yeah, hopefully that answered the first question. And then the second question around the related disease. So I think we actually, initially we also tried a version which is using multiple diseases. I think definitely improved power, but I think the technical thing, a challenge is like it's very hard to gauge the real improvement in some sense. Potentially for some disease there's related disease, then you can improve the performance, but some disease it's not, then that's very hard. So that's why in our first version we're interested just looking at the molecular kind of function which is independent of disease, and try to take in GWAS summary statistics, and then you can just learn a disease-specific thing. But I think in the future, potentially, you can integrate that.